Hi, my name is Katie Smith, and I'm a second year evolution, ecology, and biodiversity major in the College of Biological Sciences. I have been an intern in the Patricelli lab since my first quarter here at UC Davis, and I study maternal condition and differential sex allocation in greater sage-grouse. So today I'll be introducing my project and explaining my hypotheses and predictions. Then I'll go a little bit into my methods and explain how I'm doing my research, and finally I'll go over my objectives and why I'm doing this. So before we get into things, I want to establish a little bit of a conceptual background and explain what maternal condition and differential sex allocation mean and why these two variables would even be related. So first I'll explain Lucking polygyny, which is a type of mating system, then ontogeny and maternal condition and the role that they play, and finally I'll explain Trivers-Willard hypothesis, which is essentially the crux of my thesis. So polygyny is a mating system that's really defined by males mating with multiple females. The females will mate once or a few times. So if we take a bird, for example, a female bird can mate with one male, or she can mate with 100 males, but either way, she can only lay one nest worth of eggs. Males, on the other hand, can have paternity in as many nests as he can get, and therefore he's going to try to mate with as many females as possible. This makes females the limiting sex, meaning that males have to compete for females. And this can play out in multiple different ways. Males can fight for females, they can try to attract females, they can defend their females from other males, and in all these scenarios, condition is really important. So let's take a male gorilla, for example. He has to be really strong to fight other males. A peacock has to be really attractive and in great condition to grow all these feathers to attract a female. And a male elephant seal has to be really fierce and aggressive to defend his females from other males. So we see these really strong sexually selective pressures put on males to be in good condition. Lucking polygyny is a specific type of polygyny in which males will gather on a breeding ground called a lek, and they'll display for females. When all the males are gathered this way, the females can walk around and pick the highest quality male, and it's easy to compare them because they're all right there. If every female is doing this, it usually ends up where all the females mate with one male, the best male, the male in the highest condition. So it's the most extreme version of polygyny because there's huge variability in male mating success. If you're that best male, you can get hundreds of matings within a given season. If you're not that male, you may never mate in that season, maybe even in your entire life. As we also discussed, male fitness is heavily dependent on his condition in polygynous populations, and this is especially true for Lucking polygyny because it's a super strong sexual selection. Because of this, we see these very different pressures put on males and females. So males are more focused on impressing females, and they've evolved these very elaborate traits to do so. Females, on the other hand, are more focused on picking the right male and raising her brood. So because of this, we see these vast differences between the sexes in multiple different ways. Take morphological, for example. We can see vast morphological differences between this male and female peacock spider. This male has lots of colors and these strong patterns, whereas the female is quite drab. We can also see behavioral differences. So in this Wilson's bird of paradise, this male has this very elaborate dance that he's performed and rehearsed and practiced, whereas the female, she's more going from male to male, picking the best one and going to raise her brood. Finally, there's these vast physiological differences that we can observe between males and females. If you take hormones, for example, there's vastly different hormones running through males and females that are fine-tuned to their behavior. And this is precisely what I'm interested in, female physiology. Females have to make this very intense decision of who to mate with, because her mate's condition will dictate the condition of her offspring. But likewise, her own condition will also dictate the condition of her offspring for two main reasons. One is inherited genes, as her mate and her will both give their genes to their offspring, but also developmental environment is really important. So let's take a female, for example. She's in great condition. She's not very stressed out. She has a really good fat reserve because she's a great forager. She is really good at defending herself from predators, and she's built this amazing nest and she's gonna raise her broods and do a really great job. It's likely that because of inherited genes, but also this fabulous developmental environment, that our offspring are also gonna be in really great condition. So we've established this important connection between maternal condition and offspring condition. So if we think back to that male, the one that's getting hundreds of matings, the most successful male, it's likely that his mother was not an average hen, but rather a hen that looked like this, in great condition. So this brings us to Trivers-Willard hypothesis. This hypothesis states that females will invest in the offspring that is predicted to benefit most from this investment. So again, we're bringing up that male who's getting hundreds of matings. He's a male in good condition. Like we discussed, a female, and whether she's in good condition or poor condition, will still have one nest worth of eggs. So a male in good condition has a higher reproductive success than a female in good condition. If you're in poor condition, a male in poor condition is not going to mate. The high condition male is going to get all the matings. So poor condition male is going to have a fitness of 0. A poor condition female, on the other hand, is still going to have one brood. So if you're in poor condition, a female is going to have higher reproductive success than a poor condition male. Trivers-Willard hypothesis is also laid out on this graph that's easier to read. So if we looked on the x-axis of condition and y-axis of reproductive success, we can compare males and females here. So males here in blue, and you can see that a low condition male is going to have really low reproductive success. A high condition male is going to have really high reproductive success. They're very strongly correlated. On the other hand, a low condition female is going to have average reproductive success, much higher than a low condition male. A high condition female is going to have roughly the same reproductive success as a low quality female. There's not much of a difference there. So in 2005, Pike and Petrie did a study at this, and they looked at, again, maternal body condition on the x-axis, but this time the y-axis is sex proportions measured by sex ratio, measured by proportion of males. So this line here represents the average number of offspring compared males to females. And so if you have a female in poor condition, it's likely that she's going to have a female biased offspring sex ratio. If you have a female in good condition, she's going to have a male biased offspring sex ratio. So this is the importance of Trivers-Willard hypothesis, is conservation. So kakapos are a luck-breeding parrot, and they're in New Zealand, and they're endangered. So in efforts to increase population, they took a lot of birds into captivity, and they increased their survival by feeding them really high-quality diets, and they kept them all in really great condition. But because they're a luck-breeding bird, all the females were in great condition, so they all had male-biased sex ratios, which is really bad for population restoration. So this is precisely why it's so important to understand these underlying physiological mechanisms, because they're key to implementing effective conservation efforts. So this brings us to my research. I look at Trivers-Willard hypothesis in these beautiful birds, greater sage-grouse. So I hypothesize that female sage-grouse in good condition will allocate resources to male offspring. And this predicts that females with low fecal corticosterone concentration, well, I'll explain later, and high residual mass will have broods with male-biased offspring sex ratios. So my response variable is offspring sex ratio, which I'm measuring by male biases. So I'll be looking at the total number of males in a brood divided by the total number of offspring in a brood. So how do females even alter offspring sex ratio? This is what's so fascinating. It's actually unknown. We don't know how females exactly do this. So my explanatory variable is maternal condition, and I'm measuring this through body condition and through stress. So I'll be running corticosterone concentration assays on fecal samples. So corticosterone is the avian glucocorticoid, meaning it's the hormone associated with stress response. So this will give me an indication of female stress. I'll also be measuring maternal condition through residual mass, which is a calculation of weight that corrects for differences in body size. So I'm now going to be finishing my talk by just elaborating on the importance of research, and this is mostly for conservation efforts. So sage-grouse are a model organism, meaning they're used for lots of studies in sexual selection and lucking polygyny. And also they're an umbrella species, meaning that they're really important to the ecosystem, and in protecting them, we protect the other parts of the sagebrush ecosystem. They're also a species of conservation concern. So we can see in this graph here, the lighter green is historic range of their population, and the darker green is the current range. And so my research is not only going to tell us written numbers like this of number of birds lost and range loss, but future implications on population demographics. And so my research will shed a little bit of light on effective conservation efforts, like we discussed the example with the kakapo, and it can predict population demographics as they respond to environmental and anthropogenic stressors. So finally, I'd like to end my talk talking about anthropogenic stressors. In the modern world, anthropogenic pollution and stressors are becoming increasingly important, and it's absolutely critical that we understand the broader implications of our actions and the implications that they may have on overall populations. So finally, I'd like to end with acknowledgements and thank Eric Timestra and Gail Patricelli for being such wonderful advisors and helping me do all of this. I would not be here without them. Also, United States Geological Survey for providing me a fabulous data set with an amazing sample size. All the Patricelli Lab graduate students and interns for always being so supportive and teaching me so much. Kate Laskowski for always showing me so much kindness and giving me great statistical advice. Dr. Sarah Euler-McCants for teaching me a lot about sage-grouse genetics. And finally, the Han Lab for listening to my presentation and giving me advice. I'd also like to thank my friends and family for always listening to my fun facts about animals. And thank you, that's all I have for you today.